Hello, my name is Dr. Kudakasseril. Today I am going to take an important chapter in NCERT, Biotechnology, Principles and Processes. In this chapter, we are going to learn two things. One is genetic engineering, also called recombinant DNA technology, and bioprocess engineering. See, first let us look at genetic engineering. In genetic engineering, what we do is we isolate a useful gene from one organism, insert into another organism, thereby creating a new combination of DNA called recombinant DNA, short form rDNA. That is why genetic engineering is also called recombinant DNA technology. Now, let us look at bioprocess engineering. Let us take an example. You want to make human insulin from genetically modified E. coli. You culture the E. coli in a bioreactor, also called a fermentation tank. You culture the E. coli in the bioreactor, make millions of E. coli, isolate the E. coli, and from that you purify human insulin. That process is called bioprocess engineering. So, in this chapter, we are going to learn two things. One is genetic engineering and the other one is bioprocess engineering. Recombinant DNA technology was invented by two scientists, Stanley Cohen and Herbert Boyer. They created the first genetically modified organism, which was a genetically modified E. coli. So let us look at their experiment. What they did was, from Salmonella typhimurium, they isolated an antibiotic resistant gene The antibiotic was kanamycin. Okay? From the bacterium Salmonella typhimurium, they isolated an antibiotic resistance gene, or we can say kanamycin resistance gene. They put it into a plasmid. Plasmid, as we know, is a circular extrachromosomal DNA which can be used as vectors or carriers of genes. The plasmid used was called PSC 101. It was a plasmid created by Stanley Cohen. The full form is plasmid Stanley Cohen 101. So they put this gene into a plasmid. For this insertion of the gene, they had to use two enzymes. One is restriction endonuclease that cuts DNA, and DNA ligase that joins DNA. This plasmid was then introduced into E. coli. And in E. coli, this gene was expressed. In other words, we can say this E. coli was genetically modified. It was able to grow on a medium which contained the antibiotic kanamycin. So, this was the first genetic engineering experiment done by Stanley Cohen and Herbert Potter. So, they are the inventors of recombinant DNA technology. Now, let us look at the tools of genetic engineering. In order to do genetic engineering, we need two different tools. One is DNA modifying enzymes. Second one is vectors or carriers. First, let us look at DNA modifying enzymes. We need three different enzymes to do genetic engineering. One is called restriction endonuclease. Second one is DNA ligase. Third is DNA polymerase. Restriction endonuclease cuts DNA at specific locations in the DNA. DNA ligase joins two DNA pieces. DNA polymerase replicates DNA. And the second tool we need is vectors or carriers. Now let us look at restriction endonucleases. Also called molecular scissors or chemical scalpels. Before we go into the details of this enzyme, I want to make clear one thing. Nucleases are enzymes which cut DNA. They are of two types: exonucleases and endonucleases. The difference is exonuclease cuts DNA at the ends of the DNA. For example, if this is the DNA, it cuts at the ends. But endonuclease cuts the DNA inside the DNA. Exo means outside, endo means inside. So, exonuclease cuts the DNA at the ends, endonuclease cuts the DNA inside the DNA. See, I'll tell you more about restriction endonucleases. These enzymes, I told you, cut DNA at specific locations. They are produced by bacteria to cut and inactivate the DNA of viruses which attack bacteria. So it's a defense mechanism of bacteria. So that is why it is called restriction endonuclease. It restricts the growth of viruses. Now, one important thing you have to remember is restriction endonucleases cut DNA at specific locations, and these locations are palindromes. A palindrome is a word or a sentence which can be read in both directions. Example, the word Malayalam is a palindrome. You can read it in this direction and in this direction. Another example, a man, a plan, a canal, Panama. This also you can read it in both directions. Such words or sentences are called palindromes. I'll give you an example. EcoR1 is a restriction endonuclease present in E. coli that cuts the DNA at the sequence GAATTC. Guanine, adenine, adenine, thymine, cytosine. See, you can read this in both directions. That is why it is called a palindrome. So all the sites where restriction endonuclease cut DNA are palindromes. Now let us look at the naming or nomenclature of restriction endonucleases. In the example, EcoR1, E stands for genus. The genus is Escherichia. CO stands for species, which is coli. R stands for strain, which is RY13. And this one shows that it is the first enzyme discovered in E. coli. See, I want to make it very clear to you. There are more than one restriction endonuclease in one bacteria. E. coli has several restriction endonuclease. The one which was discovered first is given the Roman numeral 1. So that shows the order in which the enzymes were discovered. I want to repeat once more. E stands for genus, CO stands for species, R stands for strain, here the strain is RY13, and this shows the order in which the enzymes were discovered. Now, let us look at the classification of restriction endonucleases. They are of two types. Those that produce sticky ends and those that produce blunt ends. Okay, let us look at those that produce sticky ends. Example, EcoR1 obtained from E. coli. This has the recognition sequence GAATTC. I told you earlier, this is a palindrome. See, EcoR1 cuts the DNA at these locations here. So wherever this sequence is found, it cuts the DNA at this location. So the ends of the DNA we get looks like this. These are called sticky ends or cohesive ends. Now let us look at those enzymes that produce blunt ends. Example, SMA1 obtained from the bacterium Serratia marcescens. This has the recognition sequence CCCGGG. But it is not going to cut the DNA like this. It cuts the DNA right in the middle. So the ends of the DNA looks like this. Now these ends are called blunt ends. So there are two types of restriction and nucleases, those that produce sticky ends and those that produce blunt ends.